We are honored to have Dr. Scott Penzak give our next lecture. Dr. Penzak is department head and professor of pharmacy practice at the Harrison School of Pharmacy at Auburn University, where he has served since 2018. He earned a BS in pharmacy from Ferris State University and a Doctor of Pharmacy from Wayne State University. He then completed a pharmacology fellowship in infectious disease at the University of Arkansas. Prior to his current position at Auburn, Dr. Penzak was the director of the Clinical Pharmacokinetics Research Lab at the National Institutes of Health and held academic positions at Mercer University and the University of North Texas. His research interests include pharmacokinetics, pharmacogenetics, and drug interactions involving antiretrovirals. Please enjoy his lecture.
Hi, I'm Scott Penzak, and I'm honored to be here to talk to you today about multi-compartmental pharmacokinetic modeling. Just a couple of housekeeping announcements here. First off, I have no faculty disclosures or conflicts of interests that I need to share. And with that, here's a list of my objectives. I'm not going to bore you by reading those word for word, but they are certainly there for you to refer back to. So with that out of the way, I'd like to get started and talk about some of our typical approaches to characterizing drug disposition. And so I think in order to have a good understanding of multi-compartmental pharmacokinetics, it's important to take a step back and sort of look at the different ways that pharmacokinetic data can be assessed. And the most common way probably is the non-compartmental analysis. Most common, or certainly the simplest, most standard approach, is the non-compartmental analysis. So I'm going to take a few slides just to make sure that we all understand what non-compartmental analysis is, as well as understand the more basic type of compartmental modeling, which is the single compartment model. That way, that provides, I think, a nice landscape for us to understand multi-compartmental modeling, because then we can take a comparative look and see how multi-compartmental modeling differs from non-compartmental analysis and a single compartment model. When you can compare and contrast those two, then I think you can really appreciate what multi-compartmental modeling has to offer, as well as some of the advantages and disadvantages, and we're going to go through that. When I talk about multi-compartmental modeling, most of the time what we're going to focus on is a two-compartment model. There are certainly more complicated models than that, and if you look in the literature, you can find some extremely complicated models where there are multi-organ systems where drug is going in and out of the central nervous system, and they have compartments that might be 8, 9, or 10 compartments that are that complex. But we're going to limit ourselves to the two-compartment model because it still allows you to understand the basic premise for what multi-compartmental modeling is and what it has to offer us. So with that being said, we're going to start off with just a quick review of non-compartmental modeling, because some of the really the main parameters that we're going to talk about here are still some of the main parameters that we're going to take into account when we look at multi-compartmental modeling. So this slide is a list of definitions. It's kind of boring. So what I'm going to do is flip forward and flip back and kind of read the definition, and then we're going to look at it as it's graphically represented on the next slide, and I think that'll make the most sense. So Cmax, which is, I'm sure everyone is familiar with Cmax in a non-compartmental analysis setting. It's simply the maximum observed concentration, right? So here's your concentration. This is not log concentration, but just regular concentration. And then on the x-axis, you've got your time. So boom, highest concentration is Cmax. Easy enough, right? Next is Tmax. Well, that's simply the time to reach, move forward. That's simply the time that it takes to reach the maximum concentration. Simple enough there. All right, so now we talk about AUC 0 to last. That just means the area under the curve from time zero to the last measurable concentration here. And typically what is used is something called the linear trapezoidal method, where you break the curve into a group of trapezoids. And you can actually see right here where one of the shortcomings of a non-compartmental approach is that, see this area here between the dashed line and then this solid line, which is the actual concentration versus time curve. But when you're using a non-compartmental analysis and you draw a straight line, you're omitting part of this curve. So you can see where there's a little bit of inaccuracy that takes place there. And with multi-compartmental modeling, that's something that you hope to avoid. But here's the equation. We're not going to get into the equation. It's the addition of some concentrations multiplied by the difference in time divided by 2. So what you do is you calculate all those areas and then add them up. And then to determine this last little bit here, which gives you area under the curve from your last measured concentration out to infinity, you take your elimination rate constant, which is also called lambda z or KEL. Those names are all synonymous. You take your last concentration divided by elimination rate constant, or again, lambda z here, and that gives you that little area there, which is added to this area under the curve from 0 to the last point, and that gives you your area under the curve from time 0 to infinity. And then your elimination rate constant, that normally you'd have your natural log concentration versus time, and then you take 3 points on that line, and you estimate the absolute value of the slope there of a linear regression line, and that gives you your elimination rate constant. And so those are really your true pharmacokinetic parameters, and then you've got other parameters here that are essentially the result of calculations using the first set of parameters that I talked about. So clearance, I'm sure you're familiar, is F, or bioavailability, times the dose divided by the area under the curve. I already explained what AUC 0 to infinity is. And of course, the half-life is the natural log of 2, which is 0.693, divided by K. So there's your non-compartmental analysis. So very straightforward. And one of the key things, as we'll talk about, is this doesn't really allow you to make inferences as to what's going on in the body, whereas compartmental or multi-compartmental analysis really allows you to make inferences about what's going on in different compartments and the distributional properties of a particular drug. So what are the advantages of a non-compartmental analysis compared to, let's say, a compartmental or multi-compartmental approach? Is it simpler to apply and interpret versus compartmental modeling? Requires few assumptions about drug distribution, right? You're not really making assumptions. You're just plotting the data sort of and plugging and chugging. And it's suitable for most routine bioequivalence and exposure assessments. And we'll talk a little bit later about the FDA and what they require in terms of the type of analyses that need to be done for a new drug. The disadvantages, however, of non-compartmental methods is it's limited to observed data. It cannot simulate or extrapolate well. So these data, all those data and those parameter values that I showed you, they really can't be used to make predictions with regard to that same drug, let's say, in a different population, whereas compartmental analysis allows us to do those things. So it's less informative in terms of mechanistic insights or predictions, and it's limited usefulness with drugs that display more complex pharmacokinetics, right? Those drugs that maybe have very large volumes of distributions and prolonged terminal half-lives. They're a lot more difficult to characterize using a simple non-compartmental analysis. And so a compartmental approach really allows us to get a better look at those drugs and see how they behave in different populations. So compartmental versus non-compartmental methods. As I said, compartmental methods, they represent the body as interconnected by compartments. So this slide here is really kind of a sneak peek as to what multi-compartmental analyses looks like when you look at it in a pharmacokinetic textbook, or if you're looking at Win on Lin, for example, which is a pharmacokinetic program where you can do pharmacokinetic modeling. And when you see a two-compartment model, for example, this is what you will see quite a bit. So in fact, many of you may be familiar with the schematic that we see down here, where X sub 0 is simply the dose. 0 always means outside of the body. If you see 0, that means outside of the body. So this is the drug before it's given to the patient, right? So K0 through 1, or 0-1, is actually the absorption rate constant. So this would be for a drug that's given orally. Goes into the central compartment, usually the bloodstream or those organs that are rapidly perfused. And then for two-compartment model, the drug distributes to that second compartment and then ultimately has to distribute back to the first compartment. So you have K1 to 2. It's always in the order of where the drug is coming from and where it's going. So it goes to that second compartment. But in order to be eliminated from the body, it's got to travel back to that first compartment. So K2 to K1. And then ultimately, think of K1 to 0 as the kidneys here. So the drug is then subsequently eliminated. And we're going to look at this in more detail on another slide. But I wanted you at this point just to have a quick understanding of what a multi-compartmental, or in this case, a two-compartment model looks like schematically, because you see this quite a bit. So the compartmental model involves fitting the data to differential equations to describe drug movement. And we've got a slide coming up here that I think is going to really help bring everything together in terms of when you look at multi-compartment modeling and what's actually happening in the body with regard to that schematic with those boxes, okay? If that doesn't make sense, it will, hopefully. All right, so we're looking at the key differences in compartmental models versus non-compartmental methods. So non-compartmental methods are model independent, and they just give descriptive parameters. With compartmental modeling, it's model dependent. It's more difficult to construct and apply clinically. So that's one of the disadvantages. But one of the advantages, as we said, it gives mechanistic insight and can actually simulate and predict behavior and how these drugs behave and move throughout the body under varying conditions. And we'll talk about what some of those varying conditions might be. So, like I said, we got sort of a sneak peek ahead and we looked at the two-compartment model. So what I'd like to do now is just take a step back and simplify that for a moment and think about what a one-compartment model looks like. Okay, we talked about non-compartmental analysis, what that looks like. Well, here's compartmental analysis, but it's the simplest form, the one-compartment model. So here you have a person who has not taken the medication yet before drug, and then after dose. I don't know how well it comes out on your computer screen, but it looks like there's a bunch of little dots in here on a yellow background. So each of those dots is supposed to represent a molecule of drug. So this assumes rapid equilibrium, a one-compartment model, where the drug is taken and it rapidly distributes throughout the body. The body acts as a single uniform compartment. Kind of think of the body as this one great big bucket that has a spigot here where the drug is eliminated from. So in this case, you have the dose being given, it goes into the first compartment, the only compartment, and it's eliminated. X1 is simply the amount of drug in the body. X sub 0 is the dose, and of course K1 to 0 is the elimination rate constant. And that's what this looks like when you look at the concentration versus time curve, or actually I should say the natural log concentration versus time in a one compartment model. So one compartment, so there's only one slope of drug elimination, right? And you probably remember this equation, C2 equals C1 times e to the minus kt. So if you know the rate of decline of drug in the body and you know one concentration along the rate of decline, you can predict the future concentration simply by using this particular equation, okay? So in this part of the equation where absorption is taking place, K0 sub 0 to 1, this is where absorption is the predominantly what's occurring in the body. Then you have your Cmax here, and then elimination is primarily what is occurring. It takes over as what is predominantly occurring in the body because the absorption of the drug is over, and now the drug is being eliminated in one straight line. So you got one line, right? One slope, and you got one compartment. And if you look at this equation, e to the minus kt, in this equation, you've got one exponent. And this is a one compartment model. So that's no accident that all those things come together like that. And this just simply shows you in Microsoft Excel how one can take the equation of a line. You remember y equals mx plus b, and you look at that slope, and then you can go through and solve this equation. So now we've got, again, this equation, C2 equals C1 times e to the minus kt. We solve this equation for k, and you get 0.693 divided by k, and you come out with a half-life of 1 hour. And if you look at these data, you can see that every hour the concentration is reduced by 50%. And so this is simply meant to relate, again, to this portion of the line on a one compartment model. So that being said, let's go back and take a look at this two compartment model, which we got a peek at earlier. And now let's take a look at this in a little bit more detail, comparing and contrasting it to the one compartment model. So here, again, you've got, this is everything I mentioned before, so I'm not going to go through and all of those well, they're called micro constants, and we've got a macro constant here. So let me talk about what that is. So when we have the rate constant of drug going from the central compartment to the peripheral compartment, then from the peripheral compartment back, these are called micro constants, okay? Between the movement of drug between the compartments, those are called micro constants, whereas a macro constant, the macro constant is the elimination from the body. That's your terminal elimination. So we saw in the previous slide what a one compartment model looked like, and that's this straight line here where the drug is eliminated with there's one slope, one straight line. Now a two compartment model, you've got this steeper line up here, and you've got a dotted line, and then you've got a straight line. Well, that straight line is your terminal elimination, but this dotted line here is a distributional phase. So really that represents, as we're going to see on the next slide, that distributional phase tends to represent movement of drug from the first compartment to the second compartment. And then the terminal elimination is primarily when the drug has moved back from that second compartment to the first compartment and is being eliminated from the body. So you've got a break in the line. There are two slopes. You can clearly see one slope and another slope. And what is it? Is a two compartment model. So again, that's pretty easy to put those things together. And I would say that this is, by the way, log concentration versus time. They just labeled it concentration in the book by Sproul et al. That's I think, yeah, that on the very next slide that I have cited. So I just went ahead and corrected that if you're wondering what that is. So this next slide, I think, is really the key to understanding multi-compartmental, or specifically a two-compartment model. When I first ran across this slide, I was already out of pharmacy school. I'd finished my fellowship, and I saw this and I thought, oh my goodness, I wish someone would have showed this to me early on. This really, really helps to make sense of a two compartment model. So hopefully you'll have the same epiphany that I did. All right, so what we've got here are Roman numerals, 1, 2, 3, and 4. And then we've got 4 points, point 1, 2, 3, and 4. And then down here, we've got an explanation of what's happening at each of those times. And I've color-coded them. So what we have here, okay, this is an injection, a bolus injection. So at time 0, you assume that the drug is completely immediately present throughout the body. And that's your C sub 0, or your concentration at time 0. So this is also your Cmax, right? And let's look over here at this first point here. This is what it looks like. You've got drug filled up, and all these dots here represent drug molecules. The drug is almost completely in that central compartment. It's just starting to distribute to the second, or the peripheral compartment. And note that some of the drug is already being eliminated as well. But the primary thing that is happening, what is driving the movement, what's going to drive the movement of the drug primarily out of the central compartment, is movement into the peripheral compartment. So yes, elimination is still occurring, but it's primarily moving into this peripheral compartment. So, and that's where we are on the curve here. And this is called the distributional point of the curve. It says midway through the distribution process, drug density in the central compartment is falling. Is that right? Yes, it's falling. See, it's less dense than what it was over here, mainly due to drug distribution. Density of the drug in the peripheral compartment has not yet reached that of the central compartment. So you don't have an equilibrium yet here. The drug is still primarily moving from the central to the peripheral compartment. You're right there. So what do you think is going to happen down here at point 3? This is sort of where you have a break in the curve. And you've got, at least upon visual expression, what looks to be equal amounts of drug in the central and peripheral compartment. So let's read what we have here as far as description. Distribution equilibrium has been attained, right? So drug densities in the central and peripheral compartments are approximately equal. So at this point now you have a turning point. The major determinant of drug disappearance from the central compartment, right, is no longer movement into the peripheral compartment. Now the main reason for disappearance of the drug from the central compartment becomes the elimination process. Previously, it was determined mainly by distribution. Now it is determined by elimination from that central compartment. And so then that takes you to the terminal portion of the slope where the drug is being drained from both compartments, and peripheral compartment to the central compartment, and central out of the body at approximately the same rate. And that is your terminal slope right here. So now you've got, take a step back and say, well, this is x sub 1, this is your central compartment, this is x sub 2 from those previous schematics that we looked at. And this is the constant or log concentration versus time curve. And this is what happens, what is happening in the body at each point in this particular curve. There's one thing on here that I haven't talked about, though. This says slope beta, and you might have seen that before, where terminal elimination is referred to as T1/2 beta. That means it's the terminal elimination. So what do you think that this slope up here is called? Alpha, right? And sometimes, since we know what is happening here is primarily distribution, this is referred to as the distributional half-life. That's not really a formal term, but sort of informally you'll hear that. This is also referred to sometimes as T1/2 alpha, if you're talking about it in terms of half-life as opposed to KEL. Now here, what's going on here? This looks like somebody drew a line and back extrapolated from this slope beta, and you see a capital B here. So what is this B all about? So this, this B, as we'll see, is a back extrapolation from this terminal slope. Let's take a step back and talk about the initial slope first. Is primarily determined the distribution rate. Got a little bit ahead of myself here. Distribution half-life, or T1/2 alpha, I mentioned that on the previous slide. But the terminal slope, again, focusing on what this capital B here is, this is the negative slope of the line is referred to, like I said, as beta, and this is the elimination rate. Beta is the same as terminal elimination, or KEL, and it's, again, used to calculate T1/2 beta. Now, the y-intercept of this line, now we're finally getting to what this capital B is all about. The y-intercept of this line, in other words, you back extrapolate where it runs into the y-axis, is used in various equations for two compartment parameters. When we use a computer program, something such as Win-Non-Lin, it back extrapolates on this beta phase, and it also back extrapolates on the alpha, or the distributional phase, and it uses, and that's going to be called A, as we'll see, and basically it uses that back extrapolation in order to try to separate distribution from elimination and then make predictions as far as what's going on. So let's see what that looks like when that happens. You've got, let's see, this is just naming these points, W, X, Y, Z. So this is the actual points on the line. These are the actual concentrations where the drug's distributing. Now, if you back extrapolate from this beta slope and call these are from at the exact same time, and we'll call that W prime, X, Y, and Z prime. And then if you subtract those, you subtract W, X, Y, and Z prime from the actual concentrations, then essentially what you're doing is you're removing this elimination point. Subtraction of the extrapolated points from the corresponding actual points yields a new set of plasma concentration points for each time point. And if we plot those new points, then subsequently we generate a new line. And this is something known as the method of residuals, and it's also something that's been called curve peeling. peeling. And you can kind of see why they call it curve peeling. You're kind of removing one portion of the curve in order to isolate what is happening during the distributional process. In other words, the computer program is going to use that information to make predictions about the size of the first and second compartment, and also the rate of movement of the drug in and out of those compartments. Okay, and so that's what that looks like. You also, when you get a oops. We'll just edit right there, and I will go back here. Okay, so as we back extrapolated from the slope of line B here, we do the same thing with the alpha portion of the curve here. And again, when you look at that y-intercept, you get A. Okay, so you have this new residual line, and then the y-intercept, like I said, is called A. And from with that in mind, I ask you to just take a step back and think about how we calculate volume. And you can see another reason why it's important to back extrapolate and get this capital B and this capital A, because when you think about it, when you give a drug as a bolus and you want to find out what the volume is, that's one of the easiest ways to predict volume, because normally when you think of the concentration of a drug, right, it's just the dose of the drug over the volume. Okay, or the amount of solute that you put in a bucket of water, for example, dose divided by volume. Well, simply rearranging that equation, volume is the dose over top of A plus B. So by back extrapolating, that gives the computer program information, in other words, and how to assess the size of those respective compartments. So you've got the estimations of A, capital A and B, and alpha and beta, and they're useful for predicting volume and distribution, as well as elimination of drugs that are characterized by a two-compartment model. WinNonLin and other programs typically will ask for estimates of ranges for those parameters when they're testing and comparing models for something called goodness of fit. And we're going to talk about essentially what that means. Okay? So by exponential equation and volumes of distribution, I hinted at this earlier, right? One compartment model with the equation being C2 equals C1 times e to the minus kt. What we have here is one exponent. And so a lot of times you'll hear this as something is being called mono-exponential equation, or this graph is mono-exponential. And that's simply because the line is described by one exponent. And it differs then from a two-compartment model, which we have here our distributional phase in one compartment, and then we've got our beta phase, the second compartment. And this is a bi-exponential equation, because this is the equation for a two-compartment model. So again, two exponents, there's a break in the curve, there's two slopes, two-compartment model. Okay, so volume and distribution is something I'm not going to talk a whole lot about. It can be very complex, and again, this is one of the reasons why multi-compartmental modeling isn't something that is just done by everybody or that is used widespread therapeutically, is because it is more complicated and in general requires a computer program to generate these types of estimates and to come up with these different models. So for example, the volume in the central compartment, that's pretty simple. That's just the dose over top of A plus B, right? And we looked at that in a previous slide. But then when you start looking at the volume area, which relates to the amount of drug in the body to the concentration of drug in the plasma, in the post-absorption and post-distributional phase, now you start seeing more complicated equations. And I'm not really going to get into a lot of the complexities of those equations. As you can see, they tend to be more and more complex, especially when you start looking at predictions of a final volume at steady state, which isn't affected by changes in clearance or elimination, and is more difficult to calculate than volume in the central compartment or V sub area. And again, so you get an appreciation, really, and that's the whole point of this, is an appreciation of the difficulty of multi-compartmental modeling. But the question becomes, is that difficulty worth it? is it worth the time to go through the training, learn how to operate one of these pharmacokinetic software programs, such as WinNonLin or ADAPT software? There's a whole bunch of different ones. And I think most people would say yes, and as we'll see a little bit later, because what you gain from that, especially with drugs that have more complex pharmacokinetics, is you gain the ability to understand how that drug behaves, and you also gain the ability to make predictions. So how do you know whether your data set is best described, let's say, by a one or a two compartment model? And when I showed you those pictures earlier where you had a break in the curve, it was very clear, at least it seemed to be, whether the drug was best described by a one compartment or a two compartment model, whether or not there was a break in the curve. But let's check this out. Look at this data set. On the left and the right, the plasma concentrations are at the same spot. So you could say, well, that looks like that is best described, at least visually, by a one compartment model. It looks like one equation, C2 equals C1 times e to the minus KT. Looks like a single line there. But somebody else could say, well, wait a minute. It looks to me like that's best described by a two compartment model. It looks like you've got distribution here. This looks like there's a break in the curve, and then you have your terminal elimination. So sometimes just by looking at the data like that, it's not clear whether the drug is best described by a one or a two compartment model. So what can you do? Well, you can plug the data into both a one and a two compartment model, and then you can make comparisons as to which one gives you a better description, which one describes the data better. And then you can compare those two. And then you ask yourself, well, how do you know which one describes the data better? Well, clearly it yields, the computer program will yield different parameters that we can use to compare one with another. And what are we comparing is essentially we're comparing something called goodness of fit, because that's what these models are doing. They're trying to fit the data. So one thing that's challenging is sometimes not all subjects in a data set will behave the same way kinetically. And that can be challenging to find out if you have an outlier. Sometimes they're messing up the data, but that's something that is a little too complicated to go into a lot of detail in right now, other than to say it's important to be aware that, not every patient, or in this case, probably healthy volunteer, reads the textbook, as I've often heard said. So let's take a look at what this as comparing a couple of different models looks like from an actual data set in WinNonLin, for example. So here we've selected a two compartment model. This is kind of interesting because this is a screenshot, as you can see, and it draws the model for you. Here, they actually have a box that's labeled 0. This means inside the body. We just didn't have a box for that. So you've got your first and your second compartment, and as you can see, they just didn't have use subsets, subscripts. And they didn't put a dash between, these micro constants over here. But essentially, you have the same schematic that we showed earlier, okay? And moving here, it's asking for estimates of what? A, B, alpha, and beta. The same parameters that we talked about just a few slides earlier. And as I mentioned, it's the WinNonLin, in this particular case program, that uses estimates for those parameters to go ahead and make calculations about the size of those two compartments and the movement between the two compartments, and ultimately the elimination from that first compartment, okay? So how do you know then whether your model is a decent model or not? One of the first things that you do is look at the data. So here we have the actual concentrations, which are observed, and they are represented by these red open circles. Those are actual data. And then the model is your predicted concentrations. And what you're looking for, what you're hoping for, is that that blue line runs through those observed concentrations. And it looks, just by looking at the data, it looks like it describes the data pretty well. Another thing we do is we take those observed concentrations versus those that are predicted, which would be the difference between where that red circle is compared to where the blue line is. And what we're hoping is that they're as close to one another as possible. Well, the difference between those two, difference between observed and predicted, is something known as residuals. And then you can graph those against one another. And again, you're hoping to see a straight line. And you do correlation, and you're hoping that your R squared value is going to be as close to 1 as possible. And so up here, this is just showing you the actual data concentration. And this is for one particular patient, by the way. So you can see it says subject 1. It's telling you that is one subject, and this is a complete data for that person from time 0 through 12 hours. There's the concentrations, and then it gives you the observed concentrations versus predicted, and again, there's the residuals. So those are just the actual raw data for these graphs that you see below. Okay, so how do you actually assess that goodness of fit? We already talked about visual inspection, and we talked about looking at the residuals, the difference between predicted and observed, and the correlation between those two. There's something else called AIC, which stands for Akaike Information Criterion. And the Akaike Information Criterion, it's a measure of the relative quality of a statistical model or any given set of data. Really, all you need to remember is that AIC deals with the trade-off between goodness of fit of the model and the complexity of the model. The more parameters that the model uses, in other words, the more complex it is, you sort of take a hit for that, and it will raise the AIC to some degree. That being said, the lower the AIC, the lower the number, the better when comparing two different models. Now, the absolute number doesn't have any meaning. You could be looking at one study comparing models, and maybe AIC is in the hundreds. You can look at another model, another study that's comparing two different models. The AIC may be in the 20s and 30s or the teens, and you can't look at that AIC number between different studies. It's really only valid when you have the same data set that you're using to compare different models. And similarly, it's something called the Schwartz-Bayesian criterion, very similar to the AIC, and is also a measure of goodness of fit. And we're going to come back because goodness of fit can also be assessed by a number of many other how should I say this? Just there's a lot of information and a lot of ways to assess goodness of fit. And we're going to go over those, not really in a whole lot of detail, but I do want you to at least be familiar with what they are. And then when we do go through those, there's sort of a chart at the end that you can use to refer back to if you ever want or need to do so. So now what happens? We looked at an example where we had a pretty good fit of the data. Now, here, if we just look at these concentrations versus time, the actual or the observed versus what's predicted, we can see that something is awry here, right? We've got an observed concentration that is way up here, the predicted concentration, which is way down here. So the actual peak is higher than the predicted peak. That's the volume is certainly not accurate. The volume in the central compartment, right, is smaller than what was certainly predicted. The smaller the volume, the higher you're going to have a peak concentration, right? So you know that there is an issue with regard to that. Secondly, terminal elimination, you could see the observed concentrations is certainly looks like this would be a less steep of a slope here. So the drug is not being eliminated as quickly as what is being predicted. Actual and terminal elimination appear to be difficult to say. Without more info and without looking actually at the natural log concentration versus time plot. That's what really allows you to make an assessment of the terminal elimination of a drug. But we certainly know enough just by looking at this visually to know that this is not a very accurate model that is not well described by two compartments or that particular two compartment model anyways. So examples where the model is and isn't accurate. So predicted versus actual concentrations. This is, for example, your predicted versus your observed. This would be an example of a poor fit. All these concentrations, you can see they're off the line. You notice the differences in the predicted versus the actual concentrations. And then here you see that the observed concentrations, the similarities and the predicted versus the actual concentration. So this would be, again, an example of a better fit. But again, just looking at these residuals is just one way that we assess the goodness of fit and that we compare the models. So here, This gives you the poor fit, again, going back to what we had. We've got a poor fit versus a goodness of fit, a good fit compared to number one. So the correlation here is higher, 0.9976 versus 0.9613. That's, you would certainly expect that based upon what we saw by looking at the residuals. And here's that AIC value, 26.829 versus 47. So the lower the value, means that the fit of the data is better, and the SBC criteria is lower as well. So these are just three things that are reported in here. These in red, the square that you have here again shows your AIC, your SBC, and your correlation here. It shows those values merely at the bottom of this slide. I just went ahead and wrote those numbers again for you larger. And like I said, though, those are only really three ways to measure the goodness of fit. There's a lot more ways to do that. Quantitative data, which we talked about. This is similar to the Schwartz criteria. It's similar to AIC, but it includes a stronger penalty actually for model complexity. The number of parameters, remember I said that the more complex the model is, that will drive the AIC up. Very similar, again, with this BIC, something called the objective function value for models estimated using nonlinear mixed effect methods, and OFV minus 2 log likelihood is a key measure. So it starts getting into a lot of complexities that that really go beyond the scope of what I wanted to get into today. You've also got continuing more quantitative criteria, something called residual variance, the predicted corrected visual predicted check, the normalized prediction distribution of errors, also goodness of fits plots, goodness of fit plots, which we talked about that already, individual and population fits, shrinkage distributions. As you can see, there are many different ways to estimate model stability and robustness, precision of the parameters. In other words, their repeatability, standard error, relative standard error, covariate model evaluation, also bootstrapping, where it's a resampling technique, which is used to assess the parameter of certainty and the model stability. And then this is a summary here of really all of these different assessment types when you're looking at goodness of fit. Like I said, it can be pretty complex. Normally, what I've seen reported in a large number of studies are your correlation, difference in residuals, and your AIC. Those tend to be the most prominent ones. But the more complex the model, certainly the more of these assessment types that you'll see reported. So what are the primary purposes then of multi-compartmental modeling? It's clearly more involved than non-compartmental analysis. I think you saw that. We spent 2 slides just reviewing non-compartmental analysis. It doesn't require actually a computer program, although most people will use something like WinNonLin. However, you could actually use Microsoft Excel to do non-compartmental analysis. The very first pharmacokinetic study that I did back in the late 90s, I didn't have WinNonLin. I actually went through and calculated all the parameters by hand using Excel. Later on, when I got WinNonLin, by the way, I plugged all that data into there and got the same exact parameter values, so that made me feel good. Multi-compartmental modeling, yeah, like I said, clearly more involved. Requires a computer program, and using a one compartment model, it's more complicated than that. So why go through the trouble? And I think the answer to that should be pretty clear by what we've talked about today, and that's to better reflect drug distribution in the body. Many drugs don't distribute uniformly or instantaneously throughout the body, which is what we assume with the one compartment model, right? So that one compartment model tends to oversimplify things sometimes by assuming that the body behaves as a single uniform space or a bucket of liquid, so to speak. So by dividing the body into a central and peripheral compartment, or perhaps more compartments than that, multi-compartmental modeling offers a more realistic depiction of drug disposition. A lot of drugs are very well described by 2 and sometimes 3 compartment models. So the multi-compartment model, as I said, works better to capture complex kinetics. Some drugs exhibit multi-phase concentration time profiles. You might have a rapid distribution phase, like aminoglycosides or vancomycin, and then that's followed by a slower elimination phase. Multi-compartmental models are designed then to describe that behavior accurately by fitting the observed data to the model that includes those distinct departments for distribution and elimination. Also, the multi-compartmental modeling, if one gets a very accurate model with really good fit, it can improve the precision of pharmacokinetic parameters. Multi-compartment models yield more accurate estimates of volume and distribution in each of the compartments, intercompartmental clearance, right, or the elimination between the different compartments, those micro constants, also half-lives associated with the distribution and elimination. Such parameters are critical when scaling doses, adjusting regimens, or predicting drug interactions. So the more complicated a drug is with regard to its pharmacokinetics, multi-compartmental modeling really can help you learn more about that drug. And this can be very useful if it's a brand new compound that is not come to market yet, but you want to know as much about that drug as possible. And this can come in very handy when trying to predict certain types of toxicities. Let's say a drug has a very, very latent elimination phase, a gamma elimination, as to what it can be called. And so maybe that means the drug hangs out in renal tubular cells. Maybe it hangs out in hepatocytes. And that could be important in terms of toxicity for that particular drug. So multi-compartmental modeling really allows us to make inferences about the drug, and also to make then predictions about how that drug is going to behave in patients that may have different physiological properties. And I think this is going to come up in one of the other slides, but yeah, the second one here, personalizing therapy. If it's somebody who is obese or pregnant, somebody with renal or hepatic dysfunction, or has a particular genetic makeup where they're a hypermetabolizer or a poor metabolizer, understanding the complexities of how the drug behaves in a multi-compartmental setting, using that type of approach can help you to really understand how the drug is going to behave and make better predictions with regard to dosing in specific populations. So these are some of the important therapeutic applications that multi-compartmental modeling can have. Okay, it can help determine dose and dosing frequency by simulating drug behavior under different dosing scenarios. I mentioned what some of those are, and this could be critical for narrow therapeutic index drugs, digoxin aminoglycosides, and of course other drugs that are newly coming on the market that may have a narrow therapeutic range, where under or overdosing may have a very serious consequence. And the better you understand how that drug behaves, the better you're going to be able to choose a drug that's safe and effective for the patient. As you might imagine, designing controlled release formulations is very important to understand using multi-compartmental models, how the drug behaves, and to predict how changes in drug formulation affect the absorption, distribution, and elimination, and supports pharmaceutical development of extended release or targeted delivery drugs. And if you have a targeted delivery drug, or a drug that has a very complex controlled release formulation, it's simply going to be too complicated to be easily described by a non-compartmental analysis approach. It's going to need something that, in all likelihood, is going to require maybe even more than two compartments to accurately describe how the drug behaves in the body. And predicting drug accumulation and toxicity, I already touched on that, especially when you have a drug, again, with maybe one of those really long half-lives like amiodarone. So what about the FDA? What do they require if you have a brand new drug, you're bringing it to market, or you're doing a bioequivalent study, or you're doing a drug-drug interaction study? The FDA does not mandate compartmental modeling for all drugs. Non-compartmental analysis is generally sufficient for straightforward bioavailability and bioequivalent studies, and also first in human and just routine pharmacokinetic assessments. Maybe you're looking at the influence of a known inhibitor or a known inducer on the pharmacokinetics of a drug, and you just want to see if it changes the area under the curve, and you're going to measure difference in the geometric mean ratio, and you're going to look at those and see whether there's a significant interaction. In those situations, non-compartmental analysis generally will work just fine, but compartmental modeling is often required when you're developing population PK models, conducting physiologically based PK modeling, also supporting complex dose exposure response relationships, and informing labeling for special populations. And we kind of talked about that a little bit earlier. Some of the disadvantages, well, we know those, again, I've already said several times, it's more complex. Well, you get more information, that's just, there's no such thing as a free lunch, right? You have to put forth more effort and it takes a little bit more time, but what you get out and the information you gain and how you're able to use it expands, okay? Overfitting, that's something that's kind of interesting. You may overfit or misrepresent data if the incorrect model is chosen. And that's one of the reasons why the AIC and SBC criteria, or BIC criteria, I guess is, increases if you have too many parameters. And this means that overfitting in PK modeling means that the model is too tailored to that specific data set. So it's picking up and responding to noise in that data set. It's overfitting it. So what that does is, yeah, it makes it absolutely perfect for that data set, but it's kind of being led astray by that data set, if you will, in that it reduces its usefulness for prediction or generalization to broader clinical scenarios. So that's something to be aware of, and that's something that really, the more that you do fitting and work with data sets, the more likely you are to be aware of a situation like that where overfitting is a potential problem. Also, it assumes certain structural and kinetic behaviors that may not be biologically accurate. All right, so in summary, multi-compartmental modeling provides a more physiologically realistic and mathematically flexible framework for analyzing drug disposition. This is basically a summary of what I've talked about before. Therapeutically, it supports safer, more effective, and more personalized treatment. It's also very useful and can help to enhance drug development and really underpins rational pharmacotherapy. So like I stated earlier, it requires more effort, but that extra effort is worth it because you get more information back and it yields insights that non-compartmental analysis and a simple one-compartment model simply can't provide. So that extra effort then is subsequently worthwhile because you can certainly do a lot more and predict a lot more, especially for critical or complex drug therapies. So with that in mind, I hope you have a better understanding of multi-compartmental modeling, and I appreciate your attention here today. So thank you all.